And coming to the steps of the PCR. So mainly there are three major steps involved in the polymerase chain reaction. So first one is denaturation. So as I said before, denaturation, it involves the breaking down of hydrogen bonds from that DNA molecule. So in high temperature only the denature of the DNA strands will applicable. So denaturation and annealing. Annealing is the process in which the strands of DNA which is binding with some of the primers to be added in the annealing step, and in the extension step with the use of DNA polymerase enzyme, the primers has to be extended from 5' to 3' connections and the new DNA strands will formed. So coming to the denaturation, we could apply 94 to 98 degrees Celsius to separate the DNA strand. So as we though DNA is the double-stranded molecule, while apply 94 to 98 degrees Celsius to that double strand, so double strand molecules of DNA will get denatured into single strand and single strand. Okay, so this is last for 15 to 30 seconds in PCR. And coming to the annealing, so there are two single strand of DNA. So annealing of primer in the sense ATGC, adenine, guanine, thymine, cytosine are all attached to that one strand and attached to the another strand, and this lasts for 30 seconds. And coming to the extension, the DNA polymerase enzyme will bind and extend the primers towards the long DNA strand, and this cycle is repeated for about 25 to 40 cycles at least. So this step selectively amplify the targeted DNA sequence and defined by the specifications. So you could see this is the process which is happening inside the PCR tube which is inside the thermal cycler. So first one, the DNA template with the sequence of interest. So this is our first one is the our template DNA, which is original DNA. So you could see in first step the double strand of DNA molecule will get separated into single strand and single strand in denaturation step, and followed by the annealing step. You could see a pink color primer is attaching to both of the DNA strands. So one is up and one is down. You could see the attaching of primer in both the strands. And extension, the DNA polymerase will extend that primers towards the last. So as I said before, nearly 30 cycles has been replicated for that amplification of the DNA. So in second cycle, 1 DNA composed to 2 DNA, and in third cycle 2 DNA composed to 4 DNA. So likewise 40-50 cycles or millions copies of DNA is released or produced, it is amplified in that PCR tube. So this is the process which is overall happening in this. So coming to the procedure, we have certain steps involved in that PCR procedure. So first one is the preparation of the reaction mixture. So this reaction mixture is prepared only in the sterile environment to avoid unwanted contamination, and this PCR typically needs only 25 to 50 mol. So you imagine how minute a quantity is sufficient for the amplification of the DNA. So 25 to 50 microliter of the reaction mixture is needed for the PCR reactions to be carried out. And first one, we have to add the template DNA, which is the main source, that is 1 to 100 nanogram, and forward and reverse primers. So for the annealing steps to be carried out, we have to add both type of forward primer and reverse primer, and both can be added 0.1 to 1 μm each, and dinucleotide phosphate 200 mol, and tag polymerase 0.5 to 2.5 units, and PCR buffer in 1x concentration. And we want to add the MgCl2, magnesium chloride, and finally we want to add the sterile water to adjust the final volume up to 25 to 50 mol. So after the reaction mixture has to be prepared, it has to be transferred or loaded into the thermal cycler, which I have seen already. So in thermal cycler, so this thermal cycler consists of 96 well plate in that machine. So a thin walled PCR tube have the capability to insert in that 96 well plate, and it is loaded into the thermal cycler, and that is the programmable machine. We will change accordingly the time and temperature as per our wish, denaturation, annealing and extension, time and temperature. So setting the cyclic condition, it involves the setting of respective time and temperature. So denaturation in this is 94 degrees Celsius for 30 seconds. Annealing, it is 50 to 60 degrees Celsius for 30 seconds, and extension 72 degrees Celsius for 1 minute. So we can set accordingly in that PCR machine, and after setting the parameters, the machine is able to run, and thermal cycler automates the cycling process, ensuring the temperature and time transition. So after completion, the amplified DNA is ready for the visualization.